Myself, Mr. Mate HT, Department of Botany, RBNB College, Sri Rampur. So today we are going to study the T.Y.B.Sc Botany, paper first, BO 331, that is the cryptogamic botany. And from the cryptogamic botany, the topic number 4, that is the fungi. So first, the definition of the fungi. Now fungi are generally the cryptogamic plants in which they do not have the flower. So they reproduce with the help of a spore. So such type of plants are called as the cryptogamic plant. Now they defined as, fungi are defined as achlorophyllous eukaryotic thallophytes. Achlorophyllous, that means that they do not have the chlorophyll pigment. So their mode of nutrition is heterotrophic in mode of nutrition. And eukaryotic, that means they have a nucleus with a nuclear membrane. And third character, that is a thallophyte, that means that they do not have root, stem and lip-like structure. So such type of a plant body in which there is no root, stem and lip-like structures are called as the thallus. So the definition of the fungi is achlorophyllous eukaryotic thallophyte. Next, that is the general characters. General characters of fungi. So first, that is the habitat. Cosmopolitan, saprophytic, parasitic are there, and symbiotic. So this is about the habitat, that means where these fungi are found, and they are cosmopolitan in distribution. They are found everywhere in each and every type of the climatic condition. So they are called as the cosmopolitan in distribution or in occurrence. Now some fungi are saprophytic, that means they are found on the dead remaining of the plants and animals. So such type of fungi are called as the saprophytic fungi. Some fungi are parasitic, that means they are parasite in or on the surface of the plant body, and such a type of fungi are called as the parasitic fungi. And some fungi forms a symbiotic relationship with alga, for example lichen. So this is about the habitat. Next, that is the habit. That is the structure. This is unicellular. For example, Saccharomyces. This is a cynocytic. It is found in Rhizopus. These are nuclei. This is a monokaryotic mycelium. Central pore, septate mycelium. It is found in Puccinia. It is also found in Puccinia. It is a dikaryotic, it is a monokaryotic. And this is the dikaryotic. And last, that is the irregular plasmodium is there. These are number of nuclei. This is called as the plasmodium. So these are the different types of the shapes and size of the fungi which are found in the fungi. So number one, that is the unicellular. That means it is a non-mycelial. It is formed by a single cell only. For example, that is the Saccharomyces. In the Saccharomyces, which is also called as the yeast, the plant body, the fungus is formed by only a single cell. Now there are some mycelial structures are also found in fungi. For example, in Rhizopus, there is the cynocytic fungi. That means the fungi, they do not have the central pore, or they do not have the central pore, central septa. They do not have the central septa, and such type of mycelium is called as the cynocytic mycelium, in which there is no septa. It is called as the aseptate mycelium, or it is also called as the cynocytic mycelium, in which large number of nuclei are suspended in the cytoplasm, and it is found, for example, in Rhizopus, it is called as the cynocytic type of mycelium. Now there is another branched mycelium. Here there are the septa is there. So these are called as the septa, or they are also called as the cross walls. In between two cells there is a septa, and for example in this Puccinia, in the septa there is a central pore. Now this mycelium is a branched, and it is called as the septate mycelium. And here in Puccinia, in each cell there is a single nucleus. So such a type of cell or this mycelium is called as the monokaryotic mycelium. So in fungi, in Puccinia, there is another type of mycelium is also there. In each cell there are the two nuclei, and from these two nuclei, one nucleus is of the plus strain and another nucleus is of the minus strain. Now it is also septate mycelium, and as there are the two nuclei are found in each cell, then that mycelium is called as the dikaryotic mycelium, and it is also found in the Puccinia. So in Puccinia there are both the type of mycelia are there, that is the monokaryotic and that is the dikaryotic mycelium. But in another fungi, for example in mushroom, there is a different structure is there. Now this is the stipe, and above this stipe there is umbrella like structure which is called like the mycelium. But both in this stipe and this pileus, there is only the dikaryotic mycelium is there. And last, that is the plasmodium. Here in this plasmodium, it is amoeboid in shape. That means it is irregular in the shape. It do not have any proper shape. And number of nuclei are suspended in the naked cytoplasm. That means it do not also have the cell wall. So such type of plasmodium or the fungal body is found in the lower fungi. So this is all about the structure of the fungi. Now in fungi, there is the most important character of the fungal cell, that is the fungal cell wall content. Fungal cell wall content. Generally in the fungal cell wall there is one content which is called as the fungal cellulose, and it is called as the chitin. This is the characteristic feature which is found in the fungi. The next character, fungal nutrition. They are achlorophyllous. That means they do not prepare their own food material because they do not have the chlorophyll pigment. So such a type is called as the heterotrophic in mode of nutrition. It is called as the heterotrophic mode of nutrition, and on the basis of this there are the three main types of the fungi. For example, saprophytic. Saprophytic one, parasitic is there, and symbiotic is there. So these are the three different types found in the fungi on the basis of their nutrition. Now from this number one, that is the saprophytic. Here the fungi take their food material from the dead organic remains of the plants and animal. Second type, that is the parasitic fungi. There are number of fungi which acts as a parasite. They live in or on the surface of the plant and animal, and they causes the different types of the diseases to the plant or animal. So these fungi which are parasitic, they are also called as the pathogen. And third type, that is a symbiotic. There are some fungi, they form the symbiotic relationship with the alga, for example lichen. Now result of food material. Fungi stores different types of the reservoir food material, and this is the one biochemical character of the fungi which is important for the preparation of the classification. And these reservoir food materials are, for example, starch is there, protein, fat and glycogen. These are the different types of the reservoir food material found in the different fungi. Now next character, that is the fungal growth. Now generally number of the fungi, they are mycelial in type. Their plant structure, their body structure is a mycelial, just like this. It is branched or unbranched. Now when it breaks into a number of fragments, then each fragment is called as the hypha. And this hypha, or if there is the mycelium, it continuously grows at its tip. So there is a continuous growth at the tip of the fungi or at the tip of the mycelia. Now nuclear status in the fungi. Nuclear status in fungi. This is a monokaryotic mycelium. This is a dikaryotic mycelium. So this is the different status of the nuclei according to their distribution. So in number 1, the mycelia or fungi is called as the cynocytic. That means there is no cross wall in the mycelia or hypha. That means there are a number of nuclei, they are simply suspended in the common cytoplasm. So such type of fungi or mycelium is called as the cynocytic mycelium, in which a large number of nuclei are suspended in the common cytoplasm. Now second type, that is the monokaryotic mycelium. Here in each cell there is a single nucleus is present in each cell. So such type of mycelium is called as the monokaryotic mycelium. And there is the last type, that is the dikaryotic mycelium, in which in each cell there are the 2 nuclei are present. Sometimes they are opposite strain. From this, one is of the plus strain and another is of the minus strain. So that means in each cell, as there are the 2 nuclei are present, then such a type of a cell is called as the dikaryon or dikaryotic cell, and that mycelium is called as the dikaryotic mycelium. Now next point, that is the flagella. They are generally found in lower fungi. Their number, type and position. Now this flagella, there are different types of the flagella are found in the fungi, and their number is different in a different fungi. Their type is also different in different fungi, and their position is also different in different fungi. So this is the most important character for the preparation of the classification. Now the types of the fungi, the types of the flagella. So this is the whiplash type of a flagella. Only one flagella is there. These are the heterocon type of a flagella. These are heterocont flagella. Isocont flagella. Alright, this is a whiplash flagella. And this is a tinsel type of a flagella. So in fungi, generally in lower fungi, there are different types of the flagella are there. Their number, their position is also different in a different fungi. So here in this number 1 figure, there is only a single flagella and it is posteriorly, that means it is toward the lower side. And here there are the 2 flagella and they are anteriorly inserted. They are called as the heterocont flagella. Hetero, that is the different, and cont, that is the thread-like structure. That means here in this figure, one flagella is small and another flagella is long, and both are of the whiplash type. Now here in this figure, there are again the 2 different types of the flagella. One is a tinsel type and another is a whiplash type. Tinsel type is very small as compared to the whiplash. Whiplash is very long. Now here the flagella are laterally inserted, and again there are the 2 different types of the flagella. This one is the tinsel type of a flagella and this long flagella is the whiplash type of a flagella. Now here both the flagella, they are anteriorly inserted and they are same in length. So such type of a flagella are called as the isocont flagella. Iso, that is the same, cont, that is the thread-like structure. That means their length of both the flagella is the same. So they are called as the isocont type of a flagella. Okay, thank you.